Hello, everyone. Welcome to the CyVerse webinar series for spring 2022. I'm Tina Lee, CyVerse's User Engagement Officer. Today's webinar will be presented by Ruchika on doing single-cell RNA-Seq analysis using a Seurat-based R App in CyVerse. To see what other webinars we'll be presenting this spring, please visit our website webinars page at www.cyvers.org/webinars. Many of you already use CyVerse and know that we are an NSF-funded cyber infrastructure project whose mission is to build and deploy a national computational research infrastructure for life sciences. This free webinar series is designed to fulfill an important part of that mission, which is to train researchers and educators how to use CyVerse's resources for computational research. Let's first take care of some housekeeping, and then Ruchika will take over. Her presentation today is about 35 to 40 minutes with time at the end for your questions and her answers. Please open the Zoom chat window to write your questions there for Ruchika, and she'll answer after her presentation. All of our webinars are recorded, and you will find this recording and other materials posted on our website webinar page in a day or so. We now have more than 60 webinars organized into playlists on useful topics on science, technology, and platform that will help you learn and do your science. So let's get started. I am pleased to introduce Ruchika, who is currently a postdoc at the Nikolic Lab at the University of Arizona, but she's working remotely from home in India, about a half day ahead, so it's nighttime for her. Ruchika got her PhD in computational biology from IIT Delhi. Her research includes applications of bioinformatics in the area of comparative RNA-Seq analysis on mouse model aging and immunobiology. She loves to cook, write poems, paint, read books, and meditate in her leisure time. Welcome, Ruchika.
Thanks, Tina, for such a warm welcome. And hi, everyone. So today I will be walking you through an app, a Seurat-based RStudio app on CyVerse. But before we get started, I would like to brief you about some basic principles of what single-cell RNA-Seq data analysis is about, like pre-processing of data, some quality checks, and some initial analysis that goes downstream of the pipeline. So this webinar will be more based towards introducing anyone who would like to do single-cell RNA analysis on their own, and they don't have much resources, but they have a ready-to-use app on CyVerse using this webinar, which will be a handy tutorial for you. For later references also, you can use the video, or you can feel free to email me at my email address if you have any further questions or suggestions for me. So let's get started. So today we'll cover, first, I will introduce what is single-cell RNA sequencing and which applications are most widely talked about and where we can use this analysis. And then I'll introduce you briefly to Seurat and CyVerse. And then we will do some basic steps of analysis. As I mentioned, that the single cell is a high dimensional data set where we can just explore the data depending on the questions we are asking, but some preliminary analysis would be common irrespective of the question that you will be asking. And the common steps that I will be covering today would be data pre-processing, how to get started with your data once you have the data, how to do quality check on your own, and how to get ready with the interpretation of data on your own using this app. And then I'll show you how to launch the app on CyVerse, and followed by a demonstration using already set program commands and a data that I have already uploaded in a public forum so that you can also try at your own, followed by question answers at the end of the webinar. So what is single-cell RNA sequencing and why is this useful? So when we see in compared to the bulk RNA-Seq, where we see the overall population, it gives us a broad overview of the population in bulk RNA-Seq. But in case of single cell, we try to identify what are the differences in the cells So here is a chance where we can try and identify if there exist any rare populations of cell types. And there have been many known publications recently which have claimed to discover new set of populations, subtypes of dendritic cells, macrophages, and progenitor cells with the help of single-cell RNA-seq analysis. Also, in case of bulk RNA-seq analysis, you will get the gene expression as an averaged output from all the cells. In case of single cell, that will not be the case. For each gene expression, for each cell type, you will have the gene expression by every gene individually counted, and you get to see the minor changes as well inside the data sample. However, let's take this example where we have taken bulk RNA-seq analysis and we wanted to check whether the gene expression of gene X changes before and after the perturbation. And we see in bulk expression where the gene expression gets averaged out, we don't see any change before and after. However, when we do single cell RNA sequence analysis and we have different cell types clustered, we just see that cell type B after perturbation does show some low expression. So this downfall in expression of gene X actually tells us that single cell can help us identify cell type specific changes in transcriptome. Not only that, the single cell is also making its way in discovering of precision drugs. Like in cancer medicine, people have started discovering precision drugs for breast cancer and some of the ovarian cancers, wherein they have tried to identify what is the heterogeneity in the cancerous cells, what are the CTCs, that is the circulatory tumor causing cells, which are heterogeneous in the patients, and they try to do a precision drug modeling. Also, in case of neuroinflammatory diseases, there are some neuromedicines which have been recently talked about as a precision drug for neurodegenerative disorders. Not only that, we also can do frequency check for different cell types at certain specific intervals, also before and after a treatment is given. So characterization and frequency of cell type is another possibility, along with differential splicing between populations, identifying allele-specific expressions, and also to identify the genes that drive a process. And on a whole, when we want to check how the development process goes on, we can see the trajectories of the single cells that we have captured, and we can use them for the trajectory analysis to determine the developmental processes. So the story of single cell started in 2009, where first single cell was sequenced, and that time only one cell was sequenced, and now it has started to grow exponentially. Now we are dealing with millions of cells being sequenced. So 10x genomics is also taking care of more high throughput, high throughput in terms of cells, in terms of reads, and also it gives us new techniques, not only to do single cell RNA-seq analysis, but also to do DNA-seq analysis. And also there are latest techniques like ATAC-seq, which helps in analyzing and measuring the chromatin accessibility of the cells. And there is also spatial transcriptomics, which identifies the spatial arrangement of and the movement of cells under specific conditions. And there is the single cell DART technique, which is single cell deep learning ATAC-seq and RNA trajectory-seq analysis. This is the new technique that has emerged, which combines both the ATAC and this, and it helps in recently a publication came which claimed to identify, just using the methylated sites, how the cell types differ and which stage of cell cycle they are in. So there are many possibilities and the questions that you can ask using the same technique. For this webinar, I'm keeping it basic so that everyone can get started and on board with single cell, but definitely there is more to explore, and please feel free to contact me and we can maybe have another seminar webinar where we can talk more about specific questions. So what is Seurat? So Seurat, since we have all this data with us and we know like we want to see the data so that we can interpret it easily. So Seurat is an open source toolkit which will help us in doing all the steps that would be necessary for us to interpret the data from a matrix form to something which is more easily understandable. So this is an R package and it is designed for doing quality control checks of the data that we have. We can do analysis and exploration of single cell sequence data. And it also helps in removing the batch effects using one of the modules. We can also remove, since the samples might not be taken at the same time, or sometimes they are fresh versus frozen, or sometimes they are different samples. So all the sources of heterogeneity can be taken care. And also you can integrate different types of data. So there are diverse sets of data that are available online. Also, you can just have your own set of libraries for single cell and RNA-Seq analysis. And you might want to compare it with the previous publications, what they had done, their library prep and their chemistry and the instrument that they use to get that data might not be similar to what you might be using today. As I said, it's growing exponentially and it's every now and then there's a different version coming up. So, but Seurat, it will be very easy and handy to integrate such diverse types of data as well. And in the webinar also, I will show you how to get some free, how to get hands-on on some free data that are already available. And you can get started with them if you want to just try how to use the pipeline and how to see or get you familiar with the single cell analysis pipeline if you don't have your own library of data. Then it also helps in doing, as I mentioned, there are multi-modal functionalities of single cell where you can just analyze the spatial arrangements, you can just do the trajectory analysis to see how the, which cell cycle they are in and how they differentiate. Everything can be done on one platform. So why do we need CyVerse? So that is very important because all the single cell data which you will be getting or you will be downloading might be in gigabytes of might take gigabytes of data. Like for example, if I'm dealing with 70 patients of PBMC having PBMCs, and it is close to 500 gigabytes of data for me. So it is impossible for us to do such huge upload, download, and then use so much of RAM of our normal systems unless we are definitely working on high performance computers. It becomes very tedious for us to go back, download with some speed and do all the computational analysis on our system. So CyVerse becomes very handy in multi-functions. First, you get to have ready-made pipeline like this app right there, and where you can just feed in your data, do all the analysis on this as the steps are mentioned there. It's very interactive, and also you get to use as much of RAM space that you want. Plus, you also get help from CyVerse community or the app design app integrators. Not only that, it saves you time and a lot of work of loading the libraries and everything, because that's already taken care of and when the app was created. So I'm thankful to CyVerse for that. And let's come basically to what are the steps of analysis. So first is data pre-processing. Pre-processing means once you give your library preparation. So once you have your, let's say you are doing a gene expression analysis and you give your library preparation to someone like Novogene or somebody, and then they do data pre-processing for you, like they generate the FASTQ sequences. But you cannot use the FASTQ sequences as it is. You will download the files, separate them sample wise, and then generate a matrix. So this will be a filtered cell matrix file, which will comprise of 3 basic files, which are matrix, feature, and barcode files. So let's see how do we do that. So this is a snapshot of what I received at my end once the sequencing was done. So Novogene gave me this email saying that this is the link where you can download your sequences, like the FASTQ sequences. Here is username, here is your account name, and here is your password. All I do is I go to my computer system and I run my wget command, which is nothing to just download the file from that particular source. Here my username and password will be filled, and there is the link from which site I have to download it. And once this is downloaded, I can see that the folder is here, which contains all the FASTQ files. So within this folder, what I'm seeing are different subjects or the samples that I gave, and they have within them their FASTQ sequences. So this is just mere downloading, and you get to see that you have your samples, and within them you have your FASTQ files. From FASTQ files, since it is a 10x genomics data, I will use cell ranger count. So this command here will help me to make the FASTQ file in a matrix form. So I will come to that in the next slide, what the matrix looks like. But for now, the cell ranger count is a program that will help you to generate an output folder, whatever name you want to give here. When you give a path where your data lies and a reference genome. For example, if it is a human genome that you want to refer it to, then you give the reference genome here, and that's how it will take that into consideration and generate the matrix file. So what those output files will look like are, as I mentioned, it will be a cell, there will be several output files, but what we are interested in for any downstream analysis is this matrix, barcode, and feature file. Most of the sequencing boards like Novogene and all, they already do these steps for you if you pay them, and they give you directly these files. But just in case you don't want to shell out more dollars and you want to do it yourself, you just have to download, run one command, and there you go with the matrix files. So this is the first file that we are interested within that is the matrix.mtx file, and the contents of these file would be in the first row. Here you are seeing total number of genes that were present. Here I am trying to pinpoint on the first row what are the total number of genes that were present. So, and the next column here, the top one shows how many cells were present. And the next and the last one is basically the UMI number. So total number of UMI or the reads that were found in this sample. So you have how many genes in total, how many cells in total in this sample, and how many UMIs. And the next line onwards, you get for each gene, whether this belongs to cell number 1, cell number 2, and so on and so forth. So first, all the genes that were in the cell 1 are on the top, and likewise, once the cell 1 is covered, you will see cell 2 with the number 2 and all the list of genes that were present. So this is an identifier, and you fetch the data, like the program will fetch it for you when you do the processing on Seurat app, which I will come there in a couple of minutes. Is this identifier will actually give you an idea what is the name of this particular gene, and then which cell it belongs to, which cell number it belongs to is given by this barcode.tsv file. So every cell will have a unique barcode, 13 nucleotide base sequence, and that's how the barcode that was tagged with this gene we get to know that this gene is actually belonging to that cell. And the next to it is the UMI count. For example, for this gene, we are seeing this belongs to cell 1, and there were 4 UMIs. So 4 reads of this particular gene were there. For some of them, there were even 25. For certain genes, there might be thousands of genes or thousands of UMIs as well. So this is how the three files look like, which we will be interested in. And so we have done the data pre-processing for now. So the pre-processing was, we got the link to download. We can either click on the link, download it on our system, do wget and get it downloaded on your CyVerse or wherever you want to keep it. And then we start with the cell ranger count, get it done, and then save the matrix files. And once we are done with that, we do the quality check. How do we do the quality check? Since it is a single cell data, so anything which is not a singlet, that is multiplets, doublets, or the cells which were lysed while handling the library preparation, they are all noise to us. So only we are focused on singlets here. So only the single cells and nothing else. So how do we get to know what are the single cells and how do we remove the rest of them? So while using Seurat, there will come one point where I will show you in the demonstration as well, one graph, which we call as Boylan plot for doing quality check. In this graph, we see the N feature RNA, that is the expressed genes per cell. So here we check what are the expressed genes per cell. So generally they should be in a bin. By a bin, I mean all the cells are most likely covering this area, which I am highlighting right now, ranging from 2000 to 200. So this area is the average bin for us right now for this case. And anything above that would be the multiplets. So basically any cell that was captured, like 2 cells that were captured together, or maybe more than 2 cells that were captured together. That's the reason they were having larger number of UMIs as compared to the average cell behavior that we are seeing is lying between 200 to 2000. This is true for this case, and it varies from data to data. And once we do the demonstration, we will see that for that, the values will change, and so will it change for your data as well. So the point here is, once you find out what is the average bin, anything below and above that average, you can just remove. And how do we do that? We put a threshold of N feature greater than 200 and less than 2000 for this case. So that removes my empty cells as well as multiplets. But what about dead and broken cells? For that, I go to percentage mitochondrial transcripts and then see what is the percentage of mitochondrial genes which is outside the average bin here. So 5, if I keep as a threshold, which covers almost all of the cells which I would like to see in my further downstream analysis, I will be sure that anything above that would be dead and broken cells because they are behaving different than the average cells in this bin. So this is how we get rid of empty cells, multiplex cells, and dead and broken cells. There are other methods to remove these like scrub deads and everything, which are different depending on Python and other models, and depending on the data that you're using and the level of the stringent analysis that you want to do on your data, we can choose different modules to do more stringent quality check. But since it is a preliminary analysis and we are just getting ready to use as a beginner, so this much will definitely clean my data so that I can have downstream analysis easier for me. And I'm sure that whatever I'm doing analysis on is clean. And now let's start with the initial analysis. So we have done pre-processing. We have the matrix file. We have done quality check. We removed the doublets, multiplets, and all. Now we go for the initial analysis. So for initial analysis, first step is to do normalization. Why so? Because once I showed you the matrix file, it had different UMI counts. For different genes, there were the UMIs were ranging from 1 digit to even thousands of digits. So some genes will have UMIs at 4, 5, 10, and some will have 1,000, 2,000, 3,000 like that. So the genes which are having more UMIs should not overshadow the genes which were having less. So for that, to balance the variation, we do normalization first. And then the next step would be to do dimensionality reduction. So not all the genes within that cell will be giving me some intuitive strength about what the cell is and how do I bin them based on their properties or their biological inference. So the genes that are variable, highly variable within them will give me a more intuitive answer towards which cells are to close together, bin together, and that will not only reduce the noise for me, that will also help me in better visualizing the data and seeing that the cells are now trying to bin together based on their variable genes which they have in similar as compared to not the other ones, other cells in the sample. And these bins basically lead to clusters. And for clusters, we want to scale the data before we go for the cluster visualization, everything, so that we remove all the batch effects. So if we have the sample from X date, fresh, Y date, frozen, we want to remove whatever batch effects were there before we start with clustering, and that also helps us to visualize the data better on a UMAP. Otherwise, you will see same clusters with different on a different scale, not on the scale, but on a different place on the UMAP, although they will be similar. So it's very necessary to do scaling of data to remove batch effects. And then we do differential analysis to annotate what cells are which type, cell type. So some signatures that are unique for one cluster. So that, for example, in this heat map, you will get to know what are the enriched set of genes for one particular cluster that will help you to annotate iteratively which cell type they might have belonged to. And that's why these signatures are only specific in this bin or this cluster of cells. So coming to visualization, there are two basic visualization techniques. One is T-distributed stochastic neighbor and embedding method, which is listening. And the other one is uniform manifold approximate projections, which is UMAP. Personally, I prefer UMAP because here the distance between the clusters tells that how similar they are within themselves as compared to they are dissimilar with the ones which are different, which are clustered on like little at a distance from these clusters. For example, this cluster below 40, 45, 27, 28, 19, and 31 are close together. So this means it might be one cell subtype as compared to the others which are not so close to this one. So in clustering, for example, this is a data set which had so many clusters, and we identified through gene signatures that the green patch is the T cell type. But within the T cell type also, we have different subtypes. So how do we go to the subtypes? We take only the T cells together as one group, as a Seurat object, and then we deconvolute them based on the dimensionality reduction, and again do the UMAP clustering visualization, and then we find based on their signatures what subtypes they belong to. And this is the unique, how do we find the unique markers across all the samples, which will help us to annotate or identify this cluster is belonging to which cell type. For example, if I see NKG7 here and enzyme B, probably this is most likely to be my natural killer cells, as compared to where I see CD8 genes to be highlighted. And where I see IgG genes, probably these are the B cells. And likewise, wherever I see J chain highlighted, these are the plasmablasts. So this helps us identify based on the gene signatures to annotate which cell type they belong to. So once the annotation is done, we can go to do the differential expression genes. So how do we find the differential expression genes between two cell types of interest? We can either do based on the samples that we have, like for example, severe old versus everything else that we have. Maybe we have mild old, we have mild adult. So everything else versus severe old. So whatever is highly expressed in severe old population will pop up on the right side of the map, which is called volcano plot. And whatever is on the left side is actually not expressed most here and is expressed in the other population set. And you can also do cell type specific checks as well for differential expression. For example, in this case, CD4 cytotoxic T lymphocyte cells were checked, and from in a pool of T cells. So only the CD4 cytotoxic T lymphocyte cells in severe old, what they were expressing versus what they were, the other T cells were expressing in other samples. So you get to know the highly differential genes. You can also get the p-values, the log fold change values, and annotate accordingly depending on your own cutoff values what are the highly differential genes. And this can take you to the next level, which is the pathway analysis and further downstream analysis of if you want to do the T cell repertoire, B cell repertoire, or anything else. So what we have covered till now is basically everything that we started with: data pre-processing, quality control, and initial analysis of pipeline like normalization, dimensionality reduction, visualization of UMAP density, clustering. How do we do deconvolution and do within the cluster clustering? And then finally, differential gene expression profiles. So let's go to the live demonstration. Is my screen visible, like my CyVerse read the docs visible? Tina,
Yes.
can you help me with that? Okay.
Yes, it's visible. Thank you.
Okay, thank you. So this is the tutorial which even Tina will share with you guys after the webinar is done. Here you can see that I have taken this data set from 10x Genomics website. So if you click on this website, it will take you to the data page where I have taken the data set from, and this is freely available for anyone to use. and they also have other data as well. So among all the files that you see here, so FastQ was the input file, and after the cell range account was done, the output files were so many. And as I mentioned, we are only interested in feature cell matrix filtered file. So this will be the file that you will download in any data set that you want to use for your analysis. And if you want to take some other data set and not go with the data set that I am showing right now, or that is available in the demo tutorial, you are free to click on data set, and then you have a lot of data sets available freely on the 10x site. Also, you can alternatively go to GEO. So that is also a website where the data of gene expression is kept. And from there also you can download for each GEO the filtered matrix file. So here also you can see feature cell matrix filtered file. You can download it and you can start using it on your own, on your app, on the CyVerse account. So here you can see so many data sets are available. For now, I have taken Cell Ranger 3, and I have taken the healthy PBMC, 1K PBMC of a healthy donor version 3 of 122 cells detected. And here I have downloaded the feature cell matrix file. So if you go to this launching the Seurat app, it will take you again to the log into the discovery environment. So most of you who are already CyVerse users will already have your login, and you might be having your own account. But for some people who will be new, they can just click on login and create your own, register there and create your own login. And once it's free to use, like free of cost, you can create your login. And once you created your login, this will be your home page. So here you see data. If you click on data, you can upload your data from here, browse from your own local library, whatever files you want to upload, or you can also find out the apps from here. So now we will go to the app that we will be showing for Seurat. So you can type RStudio Seurat and it will show you version 4. That is the app that you are going to see. Okay, so this is the RStudio version 4 app. You will click. Okay. So here is the analysis. I have few running analysis going on here. Let me terminate one as it is not allowing me to launch another one. Okay, so now we will go again to apps and we will go to Seurat 4. And then here is the Seurat 4 app. And now we will go to details. Here on the details, if you see NCIBC workshop demo, if you click here and click on this save launch button, it filled in the parameters here, and click outside anywhere. It filled in the parameters here for you by default. And that will help you to get started with the app if you don't want to use your own data and you just want to get used to how to use the app and everything. Just use this demo folder. So here you can see that it has taken my username. It will be your username, and it has created an output file under the directory analysis. And by default, let it be like that. And it has given you a username and a password. So this will be used when the app will ask you to sign in. So RStudio will be username, password will be RStudio1. And here you see the input data has already taken the example data from demo folder for you to start your analysis on. So you click next, and here you keep the default parameters of minimum CPU, memory, and space disk requirement. But in case if you want to use different memory requirements based on your data, you can change it, and you can even ask them, the CyVerse support team. They're very helpful to extend the memory space if your analysis takes a little bit of more memory or space. And then here you can click on launch analysis. So all the parameters are filled by default, and you click on launch analysis. So once you click on launch analysis, it shows interactive is submitted, and here you get to know one small notification that it is submitted. Again, when you see in a little while, in a couple of seconds, you see it switch to running, and then you can either click here, access your running analysis here, or there is another more action, go to analysis. Both the ways you can reach to the app. So once you are here, you just type in RStudio, which was your username, and you type in RStudio1, which was your password, and you sign in. So here you see that you already have the folder of data, which has your input files, and in the input you have all the feature matrix file that will be required, and also the RMD, that is R markdown file. So if I click on R markdown file, it will show me all the commands that are already written for you, and you can just execute. So if you want to upload your own data, you just replace this with your own filter matrix file, or rename it with something else, your data sample 1, 2, 3, whatever, and change the path here, and just get started with here and here, and just get started with the app. For now, to save time, I have already executed all the steps here for you, so that I can just highlight on the main parts where I want your focus. So here, once the path is set in the data, you can see the demo folder in the input. And once you see what is inside the data directory, you want to check what are the contents of the feature matrix file inside the data. You can either go from here and check what is inside this feature file, which are these three files which I talked about: matrix, features, and barcodes. The very first step of what we were doing in data preprocessing, we generated these three files. So these are the three files that you uploaded here, and you downloaded the same from the site which said feature cell matrix filtered. This is the same folder, and you just uploaded it here, and this has all the three files. So let's see what the content of barcode file looks like. So within the barcode files, as I showed you on the presentation, all the barcodes will be there. So I'm just reading the top 19 barcodes. These will be unique 13 nucleotide sequences that would be there. And then let's see what is the feature CSV file look like. Here also you see the gene identifier followed by a gene name, and this was a gene expression analysis, and that's what it reads. And the matrix file, I have not read it here, but it will also have the same format of column, having how many cells were there and what do we want to see within there. And let's see how many cells do we have here. So it says we have across 1,176 cells, and we have 15,246 features, which are the genes. So these many unique genes across the samples, which are 100. So overall, we are talking about 1,176 cells. And if we want to check how do we do the quality control, so we will generate this using this plot. So this is the first step that I want to bring your notice on. So for this data, if you see, the average win here is almost close to 200 to 4,000 in this case, where the behavior of most of the cells is within this. And there are very few multiplets which are beyond 4,000. So if I have to remove singlets and multiplets, I will, for this data set, keep my threshold as 2,000 to 4,000. And also for the dead and live cells, which look like there are many in this case, 20 would be my threshold, so that whatever is below 20, which is looks like an average behavior of the cells of this sample, so about 20 would be something which is dead or broken. So then I do normalization, I find variable genes, and let's see what the top variable genes look like. So these are my top variable genes here. I get to see that these are the genes which are highly variable within the clusters that would within the cells that are there. And if you see here, I have kept the variable features since the cell size was less. I keep the N features very low. But if you have a large data set, it's a good thing that you do all genes. You do the selection method of VST, and features would be all genes. That means you are taking into account everything to find the variable genes and not just the top 2,000 gene features from each cell. And then we do scaling up of data to remove batch effect, as I mentioned, and then we do PCA. And this plot here, the alpha plot, will show what exactly the principal component looks like. And here you see that the slope changes till 20, and after that it is kind of stable. So here it is a good option to keep PC or the dimensions as 20. So here the parameter that I have kept for this example is 20. And so holds true for when I'm doing the UMAP analysis. Here the resolution you can change from 0.4 to 0.8. 0.4 to 1.2. On an average, people take 0.8. So if your data set is very less, I prefer you keep it 0.4, 0.5. If it is larger, you can keep it to 0.8 or maybe 1. And depending on how many clusters do you want to see, that will also help in keeping this resolution optimum as 0.8 or maybe 1, depending on the data that you have. And then there is this command which will plot the UMAP for you. You can also plot TISNE, but as I mentioned, I prefer UMAP, but if you want, you can just change the reduction to TISNE and run TISNE here instead of UMAP, and you can also get TISNE plot. So here we see 8 clusters showing up for the data that we have, but we don't know right now what the cell types are. So then we go and find out what are each cluster specific markers. And then within those clusters, this command will help you identify, find all markers per cluster, like plus each cluster, and that is only enriched in one cluster as compared to the other ones, and has a log4 change threshold value of at least 0.5. You can change this to 1 or maybe 2 if you do want to have a very stringent cutoff. but 0.25 is ideal if you want to just understand which clusters they belong to. And then you do a violin plot for certain gene signatures that you are confident to identify within an PBMC. So these two are B cell markers, and pretty confident that PBMC will have certain B cells, and let's see which clusters they show up in. So cluster 4 and 5 are definitely my B cell clusters. Similarly, I had few features which are NK gene, NK cell specific. So NKG7 and ERF1 are some genes which are signatures of NK. So I can identify where they are. And likewise, I can also find where are the other gene signatures on the UMAP itself. So feature plot will give me highlighted regions. The dark blue color is actually showing where the feature is abundantly found as compared to the rest of the clusters on the UMAP. So this is the same UMAP that you saw here. Just the cases here we are doing one feature or one gene at a time and highlighting it where it shows up. So based on that, I get to know these might be my platelets, these are my NK cells, these are my monocytes, and these are my CD8 cells. Also, I can do the same way for heat map and get to know or annotate where the cells, which cells are what type. Once I identified my cluster 0 is monocytes, 1 is CD4 memory cells, naive cells, NK cells, I rename them and I plot again, and I get to know that this is their positions based on the features that they are showing. So once you have all the data set ready and you want to share it with somebody, you want to save it before you move on to the next task of identifying the differential expression genes, or maybe integrate the data with some other person's data or compare it with other data set. So you save the data here, and so that before you switch to another task, you have your data ready. And this view metadata will give me a final version of how my data set now looks like. So initially when I began with, it was having just healthy N count and N feature and percentage column, but now it has my SSN res.5 and also the Seurat clusters. So basically it has the information of all the clusters that I have identified, the UMAP, and the named clusters. So you can also make a column, incorporate your own rename identities as here in metadata, which is just one command. And then you can also save that these clusters actually are these cell types. And you can also share your analysis with some of your peers. So for that, once you are here on the analysis tab on your discovery environment, here it's running. So if you click here, it gives you an option to share. So if I want to share, I can put anyone who is on CyVerse, suppose Tina, and I want to share with her. So I click on Tina and I send her, and this is done. She can also see my analysis, and she can also see what I have done, how the data looks like. So this is all about what to do and how we get started with the RNA-seq analysis. There is a lot more, as I mentioned, to do, and how do we go there would be another topic of discussion, maybe sometime later or maybe another webinar. So if you have any further questions, please feel free to ask.
Ruchika, thank you so much. We do have a bunch of questions, and I'm going to select a few of them since we are short of time for questions. So I'm going to choose ones that might be specific to running Seurat and what you've shown. Somebody asked, looks like in the analysis, they always first run PCA, then UMAP/TSNE. Why is that? Can we just do it on the original data?
So when you are doing the, as I mentioned, the protocol here itself, So you are reducing the noise before, and you are actually giving it more making it more informative for the clustering, because you are telling it that although each cell is having, let's say, 2000 genes, but only 30 or 40 genes are the ones which are highly variable, and that make the difference between one cell being different from the other, even biologically and functionally. And that inference will help us in actually doing a clustering. And that's the reason we do first PCA and then we go about clustering.
Okay, thank you. Another question, can this process be used for single nuclei RNA sequencing also?
Yes, if you have the cell data with you in the nuclei in the same format, or even if you have the FASTQ files, yes, we can use the Seurat analysis pipeline for that.
Okay, one more. How will the codes change? I'm taking it they mean in the Seurat app when having multiple samples per group.
Okay, so I understand that as you have different samples and you want to group them as a Seurat object, like you want to categorize them, that 4 or 5 bunch of samples are severe patients, some of them are mild patients or moderate patients, like that. So there is a way that you can do that in the meta-analysis, where I have shown you the metadata. You just incorporate one more column in that, and you give your reference of what group name that you would like them to be having.
Okay, and one more. How do we identify a specific cell transcriptome out of heterogeneous cell expressions? That might be a little more extensive.
I'm sorry, I lost you for a second.
The question is, how do we identify a specific cell transcriptome out of heterogeneous cell expressions? That might be a little more convoluted.
Okay, so if you want to just check one cell, you can do it using the barcode. So the barcode file is the one that can catch you just one cell. So you can just pinpoint to that barcode of particular cell that you're interested in. And that's where cell multiplexing, when we do cell sorting, basically, and we just tag oligos to one type of sorted cell. Or even if you don't want to tag or something, but you just have a particular cell in mind, you just pinpoint to that cell on UMAP or whatever, get to know its barcode, and from that barcode you can enhance your zoom out to all the UMI counts, what genes were there, what was the transcriptome look like. You can just have specific information for a particular cell. That's doable.
Okay, and then this one is really, your expertise. Someone's asking, what pathway enrichment tools would you suggest for single cell RNA-seq analyses?
So definitely go for GSEA, gene set enrichment analysis. But there are certain other tools as well, as like Metascape, which are online and free. Even GSA is online and free, but if you want to go for more precise, because they have large number of data sets to compare from, so I prefer GSA, but there are others like Metascape and all.
Okay. Ruchika, thank you so very much. We really appreciate all your expertise and sharing that with us and our community. Please join us Friday, February 25th for our normal webinar time slot. That's 10 a.m. Pacific. Our next webinar in February will be a tech preview of CyVerse Cacao, which is a multi-cloud orchestration service for researchers and educators, which eliminates the complexity of using multiple clouds. That will be presented by Edwin Skidmore, our Director of Cyber Infrastructure. We will also have another RNA-seq analysis webinar in March 25th by the lab of Dr. Bonnie LaFleur here at U of A, and she'll be, her lab folks will be showing a Python-based sequence analysis. So join us for then. Again, go to our website for what's coming up and for more information about CyVerse. And thank you, and good night, Ruchika, and thank you, everyone else. We'll see you next month.
Thank you. Thank you so much.
Appreciate it.
Sleep well. Bye now, Ruchika.
Bye-bye. Thank you so much, Tina. Let me know if you need anything else from my end.
I will. Thank you. Bye now.
Yeah. Bye-bye.